Starting YouTube.
All right.
Dr. Sanjay Singh, you're in your office?
No, I'm at home.
Oh, you're at home. Good. You're
Home sweet home.
Yeah, it appears to be like your office. So how
Yeah,
many people
because-
have joined? How many people have joined?
I have joined one polyclinic.
Okay, sir. Sir, YouTube has been started.
All right, very
Please
good.
go ahead.
Okay. So, Mr. Nanda, you can start.
Thanks, sir. Good evening, everyone. On behalf of Microbiology Society of India, Chandigarh chapter, I'd like to welcome you all to this lecture series. It is an honor to introduce our esteemed panelists and the speaker of this evening. So today we have with us Dr. Arvind M. Desmukh, the president of the Microbiology Society of India. Also, we have with us Professor Saranjit Singh, the president of the MBSI Chandigarh chapter. Professor Saranjit Singh was the former head of the Environmental Biotech Division of CSR MTech Chandigarh, and he is one of the renowned microbiologists of India. Now, without losing any time, I would like to invite Professor Saranjit Singh to introduce our speaker, Professor Javed Agrewala, professor in the Department of Biomedical Engineering, IIT Ropar, with our audience.
Thank you very much. I welcome Dr. Deshmukh, the president of MBSI, that he has given us a very good platform to do wonderful talks by eminent scientists. Today, it is my pleasant opportunity to introduce Professor Javed Naim Agrewala. He is a professor in IIT Ropar, who is no other than S.S. Bhatnagar awardee. I have known Dr. Javed over 3 decades. We used to walk and talk together in our CSIR complex in Chandigarh. We used to talk about different discoveries in science and technology, not only in India, but around the world. It was a very good walk after lunchtime we used to have every day. And the IMTech campus is so beautiful with so many flowers and trees all around. It is green, green, and very green. We attended the first leadership development program of CSIR together in HRDC, Ghaziabad. It was real fun time to attend the LDP program of CSIR, the first one in 2008. Dr. Javed did his BSc and MSc, first division from Agra University. He also did PhD from Agra University in 1986. He was the chief scientist in IMTech till March 19, 2018. Thereafter, he joined as professor in IIT Ropar. He also served as a dean in IIT Ropar. Dr. Javed received the Shanti Sharoop Bhatnagar Award in 2005 from the then Prime Minister of India, Dr. Manmohan Singh ji. And he is still displaying the snap on his mobile, the DP with Dr. Manmohan Singh, Prime Minister of India. He got the UP Science Award by the UP Governor in 2018. He got the J.C. Bose Fellow in 2018. He was INSA Fellow in 2010. He was Indian Academy of Sciences Fellow 2014. He was NASI Fellow in 2004. He has received many other awards like New Idea Fund 2000, Bill and Melinda Gates Foundation Award 2012. He has chaired many national and international conferences and gave keynote lectures and talks all over the world. Dr. Javed is editor and reviewer of many journals. He is having more than 120 publications to his credit, which have high impact factor from 5.8 to 11.1, which is a very high impact factor. He has 7 book chapters. Dr. Javed has supervised 30 students for PhD, co-supervised 4 PhD. He has supervised MTech 4 students. He has mentored postdoc fellows, 16 of them. Best of all, Dr. Javed has got 40 patents, 40 international patents, and has transferred a technology to the industry. This is cerulomycin A as an immunosuppressing agent. This technology is worth $3 million US, which is equal to 24 crore rupees Indian. He transferred the technology of cerulomycin A to Nostrom Pharmaceuticals in USA. He has got many other awards, which I am not mentioning now. I've mentioned to you the main ones. So over to you, to Dr. Javed Naim Agrawal for his talk. Javed, please.
Thank you very much, Dr. Swaranjit Singh, for a very nice introduction. So grateful to you for introducing. And you've already taken your 13 minutes, but you told me to speak for 30 minutes. So I'm going to add these 13 minutes, right? And I'm going to make my presentation full screen, right? So okay, my presentation is full screen.
Yes, very clear.
Okay, good. Okay, so today's topic of my talk is disturbing gut provokes tuberculosis, right? So as I mentioned over here, that a healthy gut is the sign of healthy life.
Yeah.
So if you remember that people who have experience of interacting with Ayurvedic or Unani doctors, the first thing they're going to ask you about your gut. What you ate and how's your motions and right. And ultimately they're going to decide on the basis of the gut what type of disease the person is suffering from. And even the Hippocrates, he said that all diseases begin in the gut. So these days the gut, it has gained very high importance because many of these diseases people have seen, they are associated with gut. And there are many papers, manuscripts which have been published. And if you see any journal these days, any high impact journal, you pick up Nature, Science, you'll find that these journals have 1 or 2 articles on gut. So whatever gut it publishes very fast and it hits in a high impact journal. So people have started believing that whatever diversity we have among ourselves, this is because the variation in the gut microbiota. And the aging, now aging is also related to the gut. Some people, they appear with age, quite young. At the same time, the person of the same age appears to be quite old. So this is being now connected that this happens because of the gut microbes which you have, right? So it depends, and the gut microbes can vary with the geography, they can vary with the diet. And the interesting part is that our intestine, it has about, 3 kilogram everybody carries in our gut, in their gut, 3 kilogram of gut microbiota, 3 kilogram of bacteria, right? 3 kilograms of whatever microorganisms they are present in everybody's gut. And there are 500 types of different species of gut, and there are about 10 to the raise of 14 microorganisms. So we have about a trillion of microorganisms which are present in our gut. And the gut, it comprises of many different type of bacteria, but largely they are dominated by lactobacilli, streptococcus, the enterobacteria, enterococcus, E. coli, bacteroids, bifidobacteria, and so on, right? So these are some of the highly dominating bacteria which we have in our gut. And different part of the gut, it has different types of bacteria. The stomach has different bacteria. Majority of the bacteria in stomach, they are lactobacillus. Whereas in the case of duodenum, the bacteria which are present in streptococcus and lactobacilli, and this the small intestine is highly populated by lactobacilli, enterococcus, bacteroids. So different parts have different, and now it is believed that people are identifying that each and every part of the body organs we have, they have some of the other types of microorganisms, and even the skin, we carry a lot of microorganisms. Okay, so these gut microbiota, they are now considered as a metabolic organ, and the reason is that they have a metabolic functions in them. And these gut microbial products, these are different types of enzymes they secrete. They secrete vitamin K, B, right? They provides short chain fatty acids like butyrate, acetate, and propionate. So all these metabolites are secreted by the gut microbes. And some of these, they are not produced by our own body system. So, and then we rely on these metabolites on the gut microbes. So that is the reason it is not being considered as a virtual organ. It is considered as a metabolic organ of our body. So gut microbial products serve as a source of signaling molecules and nutrients considered as a virtual organ. So carbohydrate, they produce carbohydrate, they help in lipid fermentation, absorption. So these are also important. They secrete certain molecules, they suppress the pathogen growth via competition for nutrients and production of antimicrobial peptides. Many of them, they secrete very, very prominent antimicrobial peptides, and they help in eliminating many pathogens. So gut microbial dysbiosis, or the disturbance in the gut microbiota, is the root of many human diseases. So gut microbiota shapes even the host immune system. So gut microbes now, or the gut microbiota, this is considered as a checkpoint for different pathogens. Example that these gut microbes, they trigger many of these cells through the TLRs, and they trigger the signaling through MyD88 dependent pathway. And this pathway ultimately leads to secretion of defensins and C type of lectins, and these lectins are quite crucial in killing the pathogens. There's a lot of murmuring from the back. Can you please mute your mic? Everybody should mute their mic. So they help in killing pathogens. Besides these gut microbiota, they also deliver signaling to different chemokine receptors, and these chemokine receptors ultimately they help in secreting beta defensins. And these beta defensins, they are responsible for active epithelial reconstitution and barrier repair, and helps in regression of pathogens. So they also secrete a lot of antimicrobial peptides, and these peptides have, immense penetration power that they can penetrate. They can kill pathogens through penetration, and ultimately leads to the lysis of these pathogens. So these symbiotin-mediated control of pathogens, these are through different type of mechanisms. So these symbiotics, they outgrow the pathogens by competing for limited nutrients and producing short-chain fatty acids and bacteriocins. So these short-chain fatty acids and byproducts, they modulate the expression of virulent genes in the pathogen. And further, what they do, these symbionts, they tune the host immune response, such as cytokines production from different set of cells. They activate macrophages, dendritic cells, neutrophils, they activate B cells, T cells, right? And they help in differentiation of even different cells like T helper cells. They help in differentiation to TS17 cell. They help in differentiation of innate lymphoid cells, Treg cells. It helps in production of different type of antibodies and antimicrobial peptides like RAG3, gamma. And secretion, they stimulate the epithelial cells of the intestine to secrete a different type of molecules. So these are all molecules which are secreted, either by these gut microbes, or they stimulate the cells of the host. So these cells, they secrete these molecules. Ultimately, they help in killing all the pathogens, right? So either through cells of the immune system, whether it's cells which are present in the pairs patches like B cells, they start producing antibodies, and T cells, they start activating different cells like macrophages, ILC, and then helping the elimination of pathogens. So if you see these days publications, gut microbiota or disturbance in gut microbes, ultimately it is associated with almost all type of diseases, whether it is as infectious diseases or it is a metabolic disease like obesity, atherosclerosis, diabetes, or IBD. Right, these are some of the diseases which are considered to be highly associated with gut. So it depends on now what type of diet you are taking. Ultimately, the diet which is good, which is a balanced diet, a prudent diet, and your lifestyle is good, a disciplined life, and immune potentiating diet you're taking. Ultimately, it raises the frequency of beneficial commensals, and it allows the decrease of the harmful or the pathogens, right? So it decreases gut permeability so that the pathogens cannot enter into the bloodstream. Further, the decreases toxemia, sepsis, pro-inflammation, and increases sensitivity to insulin, and it also increases the metabolic activity and they make healthy heart. Right. In case of if we take high-fat diets, high-sugar diets, overnutrition, right, and the life is sedentary lifestyle, use of antibiotics, it ultimately leads to the lesser frequencies, downregulate the frequency of beneficial commensals, and then harmful commensals, they increase, the frequency is expanded and ultimately leads to gut dysbiosis. And it allows the gut permeability. So these harmful bacteria, they can penetrate through gut perforation into the body, and increases the chances of endotoxemia, septicemia, inflammatory response, the insulin resistance, and adiposity, increase in diabetes, cardiovascular diseases, and so on. And the gut microbiota, it also shapes the immune system. So I'm going to talk to you later on about this. But the interesting part is that, about a few years back, there was one paper which was published in PNS, and it was a very interesting study that even if the children which are born through a normal route versus they are born through cesarean route, so they have different gut microbiota. So it was quite interesting, right? And these like babies, since they had different type of gut microbiota, so the babies which are born through normal route, they have higher frequency of lactobacillus, whereas the babies born through cesarean delivery, they have high frequency of staphylococcus and propionibacterium. And again, then when these infants, they are fed either milk, okay, or through solid food, or the food which is available in the market, they also have different gut microbiota. And once they grow, this gut microbiota, depending on the food they take, it ultimately gets altered. And again, it depends on the use. While the baby is being born, okay, or the baby is inside the womb. So if the mother is taking antibiotics, it can affect the gut microbiota, and it ultimately has an impact on the baby as well. And so the important part is that the diet of the baby, the environment, the treatment of antibiotics of these babies, breastfeeding, host genetics in an environment, they can alter the gut microbiota. Now, the important part is that the gut microbiota ultimately it can influence our health, and it can continuously affect the health from birth up to the old age. Okay, so the maternal microbiota may influence both the intrauterine environment and the postnatal health of the fetus. At birth, and about 100 microbial species, they populate the colon. And early environmental factors, like as I told you, the method of delivery, nutritional factors, breast of bottle feeding, and epigenetic factors have been implicated in the development of healthy gut, its microbial symbionts. Changes in the gut microbial composition in early life can influence the risk of developing disease later in life. So during suckling, the microbial community develops rapidly. Shifts in microbial diversity occurs throughout childhood and adult life. And in old age, there is decrease in bacteroids and an increase in firmicutes species. So the gut microbiota is important for maintaining normal physiology and energy production throughout life. So body temperature regulation, reproduction, tissue growth are energy-dependent processes that may rely in part on the gut microbial energy production. So the extrinsic environment factors, such as antibiotic use, diet, stress, disease, and injury, and the mammalian host genome continually influence the diversity and function of the gut microbiota, with implication for human health. So the disruption in the gut microbiota, which is called as dysbiosis, can lead to a variety of different diseases, like inflammatory bowel disease, colon cancer, irritable bowel syndrome, gastric ulcers, non-alcoholic fatty liver disease, obesity and metabolic syndromes, asthma, atopy, hypertension. And interestingly, even mood can be altered because of gut microbiota, mood and behavior through hormone signaling. How the mood is influenced or the behavior is influenced because the type of gut microbes we have, they affect the hormone signaling. For example, GLP-1. So the gut microbiota is also important for drug metabolism and preventing the establishment of pathogenic microbes. So gut microbiota colonizes in the initial days and weeks after birth, significantly affects the postnatal life. Okay, so the network biology approach, can aid in understanding how our gut microbiota is continuously changing in the gut environment. So the ecosystem of the microbes in the human gut operates dynamically So in order to maintain stability in the midst of constant disturbances, microbial species are continuously interchangeable via the metabolites produced by the action of gene products contained in the gut bacteria. Microbial genes and proteins and the metabolites in the gut grow from a simple structure in a yeast life, usually dominating by bifidobacteria, to a complex structure in the adults. So there is need to focus on levels corresponding to active genes, proteins, and metabolites, rather than studying the, even like microbiota only from the compositional and functional perspectives. So thus our gut microbiome is relatively stable, but it is also ever changing. And it's change, whatever metabolizes secrete, it changes, whatever metabolizer, it ultimately gives impacts on lymphoid follicles, the epithelial cell of the intestine, cells and recognition receptors, and different type of cells ultimately they influence. So the interesting part is that while we were, just the early phase when people started, doing a lot of work on gut microbiota, something we were reading, and we got very much intrigued by the papers, and we thought to write and review article. And in evening, we submitted this review article to Critical Review Microbiology. On next day in the morning when I saw this paper was accepted. So, easily, things, it's so hot topic that it is quite intriguing that people, they've started working. Many people, many groups internationally, they are working on gut microbes. And what we understood from different papers during that period, that the gut microbiota, it can prevent or it can even provoke autoimmunity. So how they provoke or prevent autoimmunity, that they can break down tolerance of autoreactive T cells. And when autoreactive T cell tolerance is broken down, ultimately it leads to autoimmunity. And how this happens? This happens because of this may happen because some of the products which are produced by the gut microbes, they may share homology or they may share epitopes with the host proteins or with our proteins. And because of this cross reactivity, it can activate the autoreactive T cells or B cells, and ultimately breaks down the tolerance, or it activates autoreactive T cells, and this is how a person gets autoimmunity. In other case, it has been observed that there are certain bacteria that leads to the activation of pathogenic Th17 cells. So these pathogenic Th17 cells, they are responsible for the activation of autoimmunity. So Th17 cells, they are very well now correlated with the autoimmunity. At this stage, there are less regulatory T cells. But in contrast, some of these gut microbes, they can increase the frequency of suppressive cells or regulatory T cells. So they suppress the activation of Th17 cells or pathogenic cells, the cells which are responsible for autoimmunity. So this is how they are controlling gut microbiota. They're controlling the activation or suppression of autoreactive T cells as well. So they also can tune our immunity. And what type of immunity they tune? Both innate and adaptive immunity. So since they have certain molecules and they can directly bind to the cells of the innate immunity, like macrophages and dendritic cells, and they can activate. So they can activate the innate immunity cells. At the same time, they can also activate the cells of the adaptive immunity, right? So because of certain molecules, these cells, they get activated, and ultimately it leads to the inflammatory response. And this inflammatory response may be beneficial to control the expansion of pathogens. At the same time, it may also be deleterious, devastating for the host in in ultimately, inflicting them with different type of diseases like inflammatory bowel disease. Okay, so these cells, the gut microbes, ultimately, whatever they are residing in our gut, they can control microbes with the pathogenic potential by competing, as I told you, for nutrients, producing antimicrobial molecules, metabolites. They modulate the function of different cells, including dendritic cells and other innate cells, of both locally and systematically, in manner that promotes the induction of effector T cells and B cells responsive to these pathogens. So when uncontrolled, this adjuvant property, okay, like directly hitting on these cells and activating them. So when uncontrolled, this adjuvant property of the gut microbes can even promote deleterious inflammatory response and ultimately the autoimmunity. Okay, so gut microbiota, and as I told you, they can very well regulate the innate immunity, and this is true since they produce many of the substances that can directly interact with PAMs, or the pathogen-associated molecular patterns. Because, I mean, they secrete something like pathogen-associated molecular patterns, and that release various metabolites that regulate intestinal and extra-intestinal immune response by interaction with various PRRs, so which are expressed on the cells of the host. And these PAMs and PRR interaction on various immune cells, it regulates host immune response and maintain homeostasis. Okay, so gut microbiota, they regulate both, as I told you, that they regulate both innate and adaptive immunity. And now the important part is, when we were reading all these manuscripts, we got intrigued because we were working on tuberculosis, and tuberculosis is a big problem for our country. And the reason is, it's not only the patients, they suffer from tuberculosis, and also the 50% of us, we appear to be healthy, but we have mycobacterium tuberculosis in our body. But this is in a latent state, it's not an active state. But whenever there's imbalance and the latent state tuberculosis bacteria comes into active stage, ultimately it inflicts with tuberculosis, right? So now what happens if we see if there is any impact of gut microbiota on tuberculosis? So whether the microbes, they can prevent, the question in our mind was whether the microbes can prevent or provoke autoimmunity, or they can also provoke tuberculosis. So for that, what we did, since there were a lot of manuscripts being published, and they showed that antibiotics, they are one of the main culprits in creating gut dysbiosis. Okay, so we thought, why not we use antibiotics? And what is the impact of antibiotics? I mean, there's already impact of antibiotics on gut. And if you disrupt the gut through antibiotics, what will be the change in the gut microbial composition, which ultimately going to provoke, or it's going to prevent from tuberculosis? So for that, what we did, we took the mouse model. We had a mouse model in our lab of tuberculosis. So in this case, what we did, we developed another model of gut dysbiosis, or gut disruption. So we took a cocktail of antibiotics, right? And these are 4 or 5 antibiotics which we use, and we fed these antibiotics to mice for 21 days. And after 21 days, we challenged these animals with live mycobacterium tuberculosis through the aerosol route. And now what we did, we rested these animals for 21 days, and then we sacrificed. And in another case, what we did, we first infected these animals with live mycobacterium tuberculosis, rested them for 21 days. And now what we did, we treated these animals with antibiotics for 21 days. So both the models we developed, in first case, we disrupted gut. In second case, we infected and then disrupted gut, right? So another model was that once you disrupt gut and challenge them, can we feed these animals with the different type of gut microbiota, or can we implant the fecal microbiota into these animals, or fecal implant or fecal transplantation we do, and then see what ultimately is the effect on these animals which have been gut has been disrupted to antibiotics and then repopulate their gut through fecal implants. Right. So interesting part, what we observed that once we treated these animals with antibiotic, there was substantial decline in the gut microbes. There was a substantial, very highly significant decline in gut microbe population. Okay, but at the same time, when we rested these animals for about 42 days, we stopped any treatment with any antibiotic, and then we did gut analysis. We found that some of these the antibiotic treated animals, they started repopulating their gut. So they start these gut microbes, they start growing, right? But the important part, which I'm going to show you, that when we did analysis, these gut microbes, they were quite different from this. So the composition, it was quite different from this. So the majority of the bacteria which were antibiotic sensitive, they got eliminated. And the gut microbes which were not sensitive to antibiotic, then they repopulated. So there was a expansion in the pool of such gut microbes that they were not eliminated. Ultimately, they populated the entire intestine. Okay, so same way, when we expose these animals with MTB, you could see, interestingly, that it also affects the gut microbiota. But then when we rest these animals, you could see that again it repopulate. So this was quite interesting, and this we could also observe in post antibiotic treatment in animals as well. So post antibiotic treatment, it could ultimately affect, and it increased start repopulating the gut of these animals. So the diversity, when we observed between these two populations, the diversity was quite unique. I mean, there was a very high diversity between these two populations, pre-antibiotic treated and post-antibiotic treatment. So there was diversity and in the population. Same time, we observed that there was a difference in the intestinal length. So animals which were not treated with antibiotics, and when the gut was disrupted, the intestine length increased. So you see here that the intestine length increased. And what we observed, why this increased, because while we disrupted the gut with antibiotic, we observed that there was the food could not be properly digested. Right. So in both animals, whether it's pre-antibiotic treatment or post-antibiotic treatment, the food was not properly digested, and since the food could not be digested properly, and now that this food got accumulated in the intestine, the cecum, and it was not moving ahead. Right. So this was not moving ahead in antibiotic treated or MTB infected in pre-antibiotic treated, and the cecum length, if you see here, it was increased. Right. So these animals, it was quite interesting to note that when these animals, they were treated with antibiotic, they showed higher level of MTB in their lungs. Upon antibiotic treatment, it increased the proliferation of TB bacteria in the lungs. So whether this is pre-treatment or post-treatment, and I would like to mention here, this is a log scale. So this difference is a huge difference. So whether pre or post, whatever treatment we give through antibiotics, it increased the frequency of live mycobacterium tuberculosis. And this we also saw in the spleen. You could see in liver, right? So what does it mean? It means that it increased the dissemination of bacteria as well. Okay, and I will tell you why there was a dissemination of bacteria into spleen or liver or the other organs. Because with antibiotic treatment, there was a perforation in the intestine. And because of the leakage, these the MTB could even penetrate. It can go through blood circulation or through lymph into spleen, into liver, and other part of the body. An interesting part of what we observed was that the antibiotic treatment increased the granuloma of increase the granuloma in the lungs. But this is only TB, and this is normal. You don't see any granulomas. So these are granulomas. These are granulomas, and you can see the staining. It shows a lot of mycobacterium over here, mycobacterium tuberculosis, right? So there are less granulomas. No granulomas is normal, but on treatment, there was substantial increase in the granulomas. So whether it was a pre-treatment or post-exposure to antibiotics, it hardly matters whether you infect first MTB or infect later on, whatever, treatment with antibiotics increases, the granuloma increased substantially. And this CFU, it increased in spleen, in liver, right? So it's post-treatment or it's pre-treatment. In both cases, there was a dissemination. That is, the bacteria started migrating to different part of the body, right? So now the important part is that which bacteria they get populated in the intestine, and they may be playing a role in increasing susceptibility to tuberculosis. So when we cultured these bacteria on treatment, there was a lot of decrease in microbial gut microbes. And on treatment antibiotics, there was a decrease in gut microbes. But this was decrease in Enterococcus, and there was increase in bifidobacteria and decrease in lactobacilli, and there was the increase in Campylobacter and also decrease in bacteroides. So that means that not all bacteria were affected, rather there were some bacteria still, they survived well, and probably these bacteria maybe are responsible for susceptibility to tuberculosis. The interesting part of what we observed that, and these animals, when we gave fecal implants, right? So when we gave fecal implant and there was a restoration in the population of bifidobacteria, it was a restoration upon fecal implants of Campylobacter. Right, so this restoration of gut microbiota, once you restore, you can see that restoration through fecal FTs, fecal transplant. What we did, we took fecal material of these animals, untreated normal animals, and then we gave through oral route to these animals which were treated with antibiotics, right? So when this fecal implant was done, we observed that there was a decrease in CFU, lung CFU. So these are animals infected with TB. These are animals infected with TB, but their gut was disrupted through antibiotics. But when we give fecal implant, we could see there was a substantial decline in the Mycobacterium tuberculosis bacteria in the lung. Same we observe in decline in the spleen. And even in post-treatment, we could see there was decline in Mycobacterium tuberculosis and in the spleen as well. And similarly, if you see here, there was a clear-cut distinction in the lung pathology. So these are the lung histopathology sections, which we are showing over here. MTB, a lot of granulomas. When they were treated with antibiotics, these granuloma further increased. So you see the size of this granuloma versus the size of a granuloma when treated with antibiotic, but they reduced upon treatment with fecal material, fecal implants, and this is pre-treatment. And if you see post-treatment, there was significant decrease in the Mycobacterium tuberculosis in the lungs as compared to antibiotic treated. So ultimately we observe that it does influence the gut microbes, disturbance will influence susceptibility to tuberculosis. But at the same time, we also observe that this antibiotic treatment, it can also affect the vaccine efficacy of BCG. So the people who are taking BCG and the children taking BCG, but at the same time they're treating with antibiotics, you see this Mycobacterium tuberculosis in the lungs of the animals. Upon treatment, the lung bacteria increased. So BCG vaccination, it did decrease the colony-forming units of Mycobacterium tuberculosis, but at the same time, when these children, if they're treated with antibiotics and they get infected, the Mycobacteria CFU is increased. And this we tested in spleen. In spleen also we could observe, not only in the lungs, but also spleen, it showed that antibiotic treatment increases mycobacterial growth. So it affects the vaccine, and why it affected the vaccine, we could see very well, it impaired the proliferation of T cells. So you see, there was substantial decline in the proliferation in the animals which were treated with antibiotics. So the percentage, it went down of T cells on antibiotic treatment, whether it was a BCG. We took two vaccines, BCG vaccine or L91 vaccination. So irrespective of what vaccine is being used, it impairs the activity of the vaccine. So whether it is T helper cell or cytotoxic T cells, both their efficacies decreases with antibiotic treatment. And you can also see that it hampers the generation of the memory T cells. So you see here, these are the T cell marker, memory marker, CD44, CD62L, CD127. So all these three markers, it ultimately showed when there's a decrease in CD44, decrease in CD127, but increase in CD62L, indicating that memory marker is decreasing. So it declined the memory generation as well. Okay, so another important thing, what we observed that if these animals, they were treated with antibiotic, they were treated with isoniazid. So isoniazid is a drug to treat tuberculosis. So the next question was, what happens if the person is being treated with isoniazid at the same time his gut gets disrupted? There's a dysbiosis in the gut. So ultimately what happens, it obstructs the clearance of mycobacteria, right? So if you see here, what we observed that when we treated antibiotics, there was a disturbance in gut microbes, right? So there was a decrease in lactobacillus. We could see a decrease in here bifido. But at the same time, we observe that when these gut microbes, they are affected, and these animals, if they are challenged with mycobacterium, they are challenged with mycobacterium treated, and they're receiving isoniazid, but the gut get disrupted. So at that instance, what we observe that it impairs the function of isoniazid. So this is MTB, untreated animals. So when you disrupt gut microbes, gut microbiota, it ultimately increases the MTB in the lungs. So this is again log scale. And when you treat these animals with isoniazid, there's a substantial decline on treatment with isoniazid on Mycobacterium tuberculosis. So isoniazid is a potent drug which is used to treat TB. So it shows decline in the CFU of these animals. But if the gut is disrupted and isoniazid treatment is given, so it increases the proliferation of Mycobacterium tuberculosis, right? So what does it mean? The gut disruptions, it hinders in the function of, in the efficacy, it reduces the efficacy of isoniazid, and this was evident even from the lung histopathology. So the lung histopathology, it shows that upon treatment, there's increasement in the granulomas. So here you see this is as good as like a normal lung, right? So this shows that the gut dysbiosis, it may have a great impact on the tuberculosis, besides other diseases which people are regularly publishing, and even it has impact on tuberculosis, can impair the function of our immune system by not allowing the immune system to efficiently clear the mycobacterium tuberculosis. So finally, I would like to acknowledge my team. So this is the team who, all of them, they participated in the study, but the main work which I talked today is done by Nargis, Nadeem, and Shika. And I would like to thank CSIR for funding. So thank you very much. And now I'm ready to take questions.
It was a very excellent talk, Dr. Javed.
Thank you very much.
I enjoyed your talk very much. Now, microbiomes are very important in the gut. Nowadays, people are doing research in treating disease by adding microbes to our gut. Yes. What do you say about this new therapy method?
Yeah, see, this is quite promising therapy which is being used, you know, and they have observed, at least in years, they have started this fecal transplants. So they're giving fecal transplants, and people are showing, at least in IBD, there's a substantial, you know, regression in the disease symptoms. Yes. And many other diseases. The problem with this fecal transplant is there's a big problem with it. The problem is because it has even, you know, some of the pathogenic bacteria which may not be beneficial. So ultimately, people sometimes they suffer from different type of diseases as well because of the overproliferation of these non-essential bacteria. So now either the person should identify the right type of bacteria, create a consortium of that bacteria. And let me tell you one important thing, that it is not a single bacteria which works. It's a consortium of bacteria that works. So either one should, one should use the beneficial bacteria, gut bacteria, which can directly be used to treat and eliminate those bacteria which may be harmful. So fecal transplant is now being gradually discouraged. So people are trying to identify the right type of bacteria. Another important thing is that beside this, people are now started working on metabolites or secondary metabolites are secreted by these bacteria, which ultimately they are responsible for decreasing or curing these diseases. So more metabolites or probiotics people are using, right? Prebiotics people are using, metabolites people are using. So that may have a very high potential in curing different types of diseases.
Right. I was reading somewhere that the microbiome in the stomach can also cause brain stroke. Have you read this report somewhere that the microbiome can, actually cause a brain stroke?
Yeah, that's very exciting. It's quite intriguing when I read the same paper, and connecting to this, there was a few more papers, and I read one more paper. And this paper, what it mentioned that there was a group. So what this group did, they took one set of animals, and they reared these animals in normal environment, right? And you may say like Indian environment, right? And second environment, what they did, they took these animals and they reared these animals in totally not a biotic environment or germ-free environment, right? So what happened, as these mice, they laid the pups, initially they were keeping these animals, right, in totally germ-free environment, not a biotic environment. Another they were keeping in a normal environment. And then when these mice that delivered pups, these pups were also being kept in such environment. Yes. And the most exciting thing which they reported was that the animals, these animals, they grow, So the animals which were developed in normal environment, they had normal brain. And the animals which were reared in germ-free environment, these animals had, very, there was no proper growth of their brain. Rather, they said it was like a rudimentary brain, and there was not normal animals. So it was quite exciting that how much,
Yes.
how much control does these microbiota it has on our body.
Yes. Very nice answer. Anybody can ask a question now. You
Yes,
can
I
ask a
have a
question to
question.
Javed.
Yeah. Hello.
Yes, yes.
Yeah. Good evening, sir. It is a really nice session and quite enlightening on the topic. So, the question is, can you comment more on FMT? Is it like clinically proven with respect to studies in India?
With respect to
Studies,
transport.
is it applicable, feasible, or is it ongoing in India or on international level, fecal microbiota transplantation in any diseased state?
Right. See, as I told you that in US, yes. In US, there are many reports, and these are published reports, that there's a fecal transplantation is being done to treat IBD. But as far as India is concerned, I'm not really aware of, fecal transplant, except I know only one case in, probably it was in Delhi, in Gangaram Hospital. They treated a person, an old person, like 78 years old person with the fecal transplantation. Right. So
Okay,
that is what, since this person, he agreed because he was having a recurrent problem of diarrhea, right?
Okay.
Or inflammation, recurrent inflammation in his intestine, and ultimately 78 years old person, and none of these treatment they were working. So ultimately he agreed, and this, despite of whatever antibiotics being used, so despite of whatever antibiotic.
I think sir got disconnected.
Yeah. Hello. Hello. Ranjit Singh, sir. Hello. Are you there? Hello.
Dr. Patankar session is completed or what?
Yeah. I'm not getting what happened.
Yeah, actually I joined late and
Yeah, because
then-
this is I think this is a Zoom meeting.
Yes,
1
yes.
hour is over.
maybe
Yes.
that
Yeah.
No, actually, there is some disconnection from IIT Ropar.
Okay. Okay.
I think the power has gone in IIT
Okay.
Ropar.
Okay.
Anyway, we have completed the meeting now. We
Okay.
have completed.
Sure, sure.
this is the
Dr.
very
Patankar, thank you for joining.
Yeah. It was a nice. I had
Yes,
one question. I had one question to ask, but it has gone regarding the.
You tell me.
Okay, sorry, there was some problem with internet.
I
Sorry.
also had one question to ask.
Yeah,
Even
Sure,
yes,
sure.
please.
I
Yes.
to ask, I think it was at the end. Fecal micro
Yeah.
and actually here Dr. Javed CFTR I sir in IMTech.
Hello.
This is Salami.
Actually, you're not fully audible. The voice is cracking in between.
Sir, may I ask one question?
Yes,
Sure.
please ask.
Sir, we are talking about the prebiotic and probiotic as such. Regarding the prebiotic, there are so many types of millets which are untapped till date, but the certain tribals are using those particular millets for the fermented foods. I worked on that, and it is giving a probiotic type of the effect. So it is a necessity to tap such type of the prebiotic sources and bring it to the common people.
Yes, definitely.
Yeah.
Definitely. See, ultimately, we have to identify. See, as I showed first slide, what we are, what we eat. What we are, what we eat. Right. So ultimately, we have to see whatever we are eating, how does it affect the health, and health, whether it is really affecting gut. So everything goes through gut, and if gut is affected, and it may be beneficial or it may be harmful. So then one ultimately has to decide what to take and what not to take. It may be very good for you to take certain food, but it may not be good for me, or the one which is good to me may not be good for you. So that is important. Yes. Anybody else?
Yeah, sir. This is
Yes.
Dr. Nagrani Mahajan here, sir. So that was a
You're from?
wonderful talk and very well designed research work that you had presented.
Thank you very much.
Yeah,
You're from?
very insightful too. I am from Latur, Maharashtra, sir.
Okay.
Yeah, I know ki Mycobacterium is a very notorious bacteria, which is very difficult when it gets into multiple drug resistance type of situation.
Yes.
So as you very well planned with coming out with a solution where you can use other microorganisms which are beneficial for replacing them with pathogenic ones.
Right.
So I just was wondering where any experiment designed to study what kind of genes were upregulated or downregulated in this process. Like when the beneficial microbes are there, what kind of genes were upregulated or downregulated in presence of that. So that could probably help to find out how our body is responding to those beneficial ones. Like
Yes.
some kind of transcriptome analysis or RNA expression levels, like what it is doing exactly with the host body.
Yeah, that's a very good question. And we have done one of my students. He has completed. He has just submitted his thesis. Exactly what you have asked the question, right? So, but this is not in context with mycobacterium tuberculosis. This is in context with aging. Right? When we did transcriptome, and
That
we
is
did
related to
that is related to aging.
aging.
Aging, yes.
Achha.
So we did observe there's a lot of changes in transcriptome, and even in genomics, you see a lot of changes in the gene level as well. So the important thing is that, yes, when we did analysis of these animals, when they keep on, growing, so it's very interesting that when they grow, as I've shown in all the slides, that the microbiota also changes. And some of the microbes, with aging, they disappear. Gradually, they disappear. And some of the microbes, with aging, they try to repopulate. They become dominant, right? And these microbes, with aging, when they dominate, some of them, they ultimately are responsible for doing all sort of immunosuppression, or they try to generate certain type of cells which may not be beneficial for the health, right? And interestingly, when we feed these animals with the fecal transplant from the young into the old one, right? So they gain the microbiota of the young animals, and also the genes are influenced. Genomics is influenced, their transcriptomic is influenced, the metabolics is influenced, proteomics is influenced. So that's quite interesting. And it'll be very interesting to see in case of tuberculosis as well, how does it influence. So the work I have that is published in case of TB as well, which I have not shown here. So we could see that some of the bacteria, especially the bifidobacteria, was very important in case of tuberculosis. So when we treated antibiotics and bifidobacteria start disappearing, and that was the event when the animal becomes, or it gives a chance at that particular instance, mycobacterium to divide, divide fast and migrate into different parts of the body in liver, spleen, right? And if you feed bifidobacteria, you can restrain mycobacterium from dividing and from dissemination.
It's majorly the role of microbial metabolites that are hindering the growth of one organism and allowing the other one to grow. And along with maybe
Dr.
the
Mahajan,
host is, yes,
Yeah,
sir.
ultimately,
Mahajan, we are running out of time. Can you
ultimately,
please write to Javed? We are running out of time.
Sir.
So we would like to
Sure,
close the
sure.
session now. You can
Sure,
write
sir.
to Javed or to me.
Sir.
We'll answer you.
Sir.
But why you are running short of
Nanda?
time? Dr. Sanjeev, why are you running short of time? It's 6:13. So everybody is at home.
Yeah, because we have some other program now, followed by yours.
Okay.
So that's why we'll again get back to you. And
Okay,
can I talk
I'll
to
give a short.
Nanda, you can give the word of
I'm
thanks.
going to give a short answer for it.
Yes. I'm sorry for that, but we have to.
Okay. Any more questions?
Conclude. Nanda?
Yes, sir, please, one question, please.
Yes.
Hello,
sir?
as I requested you, please write to us. We don't have time now.
Okay,
Sorry.
thank you.
Sorry.
Yep, thank you.
You can ask no
Nanda,
problem.
please give the word of thanks.
Okay. You can write to me. And
Please.
let me share. I don't know how to share it. I can share my email ID. My email ID is simple. You can write to Javed, 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 IMT. Since I was earlier at Institute of Microbial Technology, so IMT, javedimt@gmail.com. Right? So I can respond to all the queries.
Thank you, sir.
Is Nanda there?
Thank you very much. Thank you very much for all of the questions and the queries. I'd like to thank Dr. Arvind M. Desmukh and Professor Swaranjit Singh for organizing this beautiful session. I'd like to pay my hearted thanks to Professor Javed for giving this wonderful talk and all the participants which have been joined and make this talk successful. Thank you, everyone.
Thank you very much.
Thank you.
Thank you.
Okay, take care.
Bye.